Hello everybody, today we're going to be talking about Chapter 8, Bivariate Correlational Research. So bivariate correlations are associations that involve two variables, right? Bivariate two variables. And these can be any two variables. In this case, we've got a correlation between a score on a well-being scale and percentage of conversations rated as deep talk. And we can correlate these two variables. Particularly with bivariate correlations, we're looking at the degree of relationship among two variables. Again, for bivariate, there's two. You can have correlations among more than two, but we won't be going into that in this class. Quantitative variables is the key. So two or more quantitative variables, those are variables that are measured with numbers. So did your SAT score predict your college GPA? So every person would have a score on both SAT and college GPA, and then we would look to see if there was a pattern or relationship between those numbers. Does watching violent TV in terms of the hours of violent TV watched lead to aggressive behavior in terms of number of aggressive acts? So for every person, let's say these are children, we would see how many hours of violent TV they watch a week, how many aggressive acts they do every week, and then again we'd look for a pattern or correlation. When we are making a correlation with two quantitative variables, the statistic we run is called the Pearson product moment correlation coefficient. You probably worked on this in statistics. It's represented by a lowercase r, or it's called the Pearson R. And you are going to be going through this actually in your survey project. You'll be running a correlation, so we're going to spend some time learning how to make sense of a correlation. There are two main components. There's the strength and the direction of the correlation. The strength will be the size, and it ranges from 0 to 1, mostly a decimal, somewhere between 0 and 1. And the sign will give you the direction of the relationship, whether it be positive or negative. And of course, a correlation of 0 means there's no relationship. There's no correlation between the variables. The first thing we usually do when we have correlational data is we graph it, and the type of graph you would use would be a scatterplot to graph the relationship between your two variables, and it would also reveal any influential observations and outliers. And so we always want to graph our data first. In this case, here's years of experience versus income. So those are our two variables. Each dot represents a person in this case. So we have someone maybe who has 0 years of experience right here at their job, and they make, let's say, $20 an hour. And then someone over here has had 35 years of experience, and they make, where are we? Almost $60 an hour. So each person has a score on both of the variables, and then you plot the points and look for a pattern. In this case, we see a pattern. We see that with more years of experience, people have a higher income, right? And with less years of experience, people tend to have lower incomes. So interpreting the correlations, we're going to look at direction first, and this means when you run the correlation coefficient, it will either be a positive number or a negative number, and again, it'll be a decimal typically, so like positive 0.43 or negative 0.23. But just by looking at whether the number you get is positive or negative, you know if you have a positive or negative correlation. So what do we mean by that? A positive correlation is when the variables tend to move in the same direction, meaning as one variable goes up, the other variable goes up. When one variable goes down, the other variable goes down. If someone is above average on one variable, they tend to also be above average on the other. And if someone is below average on one variable, they tend to be below average on the other. So this would look something like this with years of education and hourly salary. Again, here's our people in our research study. This is totally made up data, as you can tell, but here we've got people, someone who's got maybe 8 years of education and they make $5 an hour. 9 years of education, they're also making about $5 an hour. And people here who have like 22 years of education are making, wow, $25 an hour in this case. But you're seeing this upward sloping line from left to right, basically, if you look at the data points and were to kind of look at the pattern, if it's an upward sloping line from left to right, you have a positive correlation, meaning that when people are low on one variable, they tend to be low on the other. Low years of education, lower hourly salary, higher years of education, higher hourly salary. And that would be if your correlation coefficient that you run in SPSS is a positive number. Now, if your correlation coefficient has a negative sign in front of it, like negative 0.42, that means the variables tend to move in opposite directions. When one variable goes up, the other goes down. If someone is above average on one variable, they tend to be below average on the other. If someone is below average on one, they tend to be above average on the other. When you graph a negative correlation, it tends to look like a downward sloping line from left to right. This is like a negative correlation. And maybe these would be the variables of if X was hours of watching TV and Y was GPA. And what we would see is when people are low on hours of watching TV, so low on this X variable, they tend to be higher on GPA. Their GPA tends to be higher. And when people are very high on one variable, like very high on hours of TV watched, they tend to be low on the other variable, which is GPA, tend to have a lower GPA. This would be a negative correlation. There's also something called a curvilinear relationship, and this is when increases in one variable are associated with both increases and decreases of the other variable. This would be, if you have a curvilinear relationship between your variables, you would be using a totally different type of correlation statistic. You wouldn't be using the Pearson R, but it still is important to know these types of variable or relationships exist. And here's one of them that is a pretty famous one in psychology, is the relationship between anxiety and test performance. Curvilinear just means it's a curve. So you can see this is a curve, upside down U. You might have two variables that are related in like a U-shaped curve or some more complicated relationship. But in this case, what do we see? We see that people who have very low anxiety tend to perform not very well on tests. And why is this? It's because they are not only not anxious, they don't really seem to care. They're not nervous, they're not worried, so they might not study. They might go out with their friends the night before the test, not get a good night's sleep, and so they don't do very well. But we also see that people with very high anxiety tend not to do very well on tests, right? These are people who have test anxiety. They get into the classroom and their mind goes blank and they can't think, and they don't do as well. But it's these people with a moderate level of anxiety that tend to perform the best on exams, right? They tend to be the ones who say, well, I am a little bit nervous. Tests are coming up. I am going to study. I'm probably not going to hang out with my friends this week. And I'm going to get a good night's sleep. And they tend to do the best. So this is a curvilinear relationship that increases in one variable, like anxiety, go along with both increases and decreases of the other variable. And of course, it's possible to have no relationship or a correlation of zero, that increases in one variable is not associated with any systematic change. Here's an example of that. You basically have a scatter plot with no pattern at all. If this variable down here was shoe size and this variable was IQ, we would see no relationship between shoe size and IQ, that people could have a small shoe size and a low IQ, and a small shoe size and a high IQ. People could have a large shoe size and a low IQ, and a large shoe size and a high IQ. There's just no relationship. So we understand the direction of the relationship just by whether the correlation coefficient is positive or negative. Now, the strength, how strongly or weakly those variables are related, has to do with the actual number. We can go from -1, meaning a perfect negative correlation, to +1, being a perfect positive correlation, 0 being no correlation. And as you move closer to 1 or to -1, so in either direction, the relationship gets stronger. Okay, so you go from a weak to a moderate to a strong to a perfect correlation. This is kind of our way that you can think about interpreting correlations, that basically, if your correlation is greater than 0.5, you have a very large correlation. Between 0.3 and 0.49, we'd call that a moderate correlation. Between 0.1 and 0.29, that's small. And less than 0.1, we wouldn't even really discuss. That's very close to 0. It's practically no correlation at all. So let's see, here are some examples of strengths of correlations. This is a correlation of +1, so a perfect positive correlation, which means the two variables are perfectly related. Every dot is perfectly aligned on this line. Here's a perfect negative correlation, this downward sloping line. A correlation of 0 with no relationship. But these are not very typical that you would get a 1, a perfect correlation. Well, it's not that unheard of to get no correlation. But what about some of these down here? Here's a correlation of 0.8. So we know it's a positive number, so that means it's a positive correlation. That's what we're seeing here. And 0.8 is pretty close to 1. This is a pretty strong correlation, that even though the dots don't fall exactly on this line, they are pretty close together. There's not a lot of variability. This is a pretty strong positive correlation. How about this? This is a correlation of 0.3. So again, it's a positive correlation. We generally have a positive relationship between these variables, but 0.3, that's a lot closer to 0. And this is a much weaker correlation. You can see there's quite a bit of variability. The dots are spread farther apart, and so it's a less strong relationship. And then a correlation of -0.2. So now we've got a negative correlation. These variables are related in this inverse way. And 0.2, again, that's pretty close to 0. So we have a lot more variability. The dots are much more spread out. But that is the relationship there. So if you run a correlation coefficient like you will in your survey project, in this case here we have 5 students and they take 2 tests, test X and test Y, and student 1 gets a score on test X and test Y, and student 2 gets a score on test X, and test Y, etc. So everybody has a score on both variables, and they're both number variables, they're both numeric variables. Your correlation or your hypothesis for this type of correlation would be something like, it is hypothesized that score on test X and score on test Y will be positively correlated. That is, the higher the score on test X, the higher the score on test Y. This should say test and not text through the whole thing. I'm sorry about that. Or you could maybe have a negative correlation being hypothesized. It is hypothesized that there will be a negative correlation between score on test X and score on test Y. That is, the higher the score on test X, the lower the score on test Y. We would run a scatter plot, and here you have a scatter plot, and if you look at this, this looks like a pretty strong positive correlation here between these two variables. Your results would look something like this. Usually point to the figure. Figure X shows that the correlation between score on test X and Y, as the figure shows, there is a strong positive correlation in which the higher the score on test X, the higher the score on test Y. And then you'll get this from your SPSS printout. Your TAs will show you how to do this, and it's also in your lab manual. R is correlation. This is your degrees of freedom, which is on your correlation table, equals 0.8. That's the correlation coefficient. Again, that's a strong positive correlation. P equals 0.001. That's less than 0.05. Remember, that's what we're always looking for the significance to be less than 0.05. And this number also would come from your SPSS printout. Or although figure X tends to show that test X and test Y are related, the correlation between the two is not significant. You might end up writing something like that if you get non-significant results. When we look at correlations, you'll want to think about moderating variables, although we will not do this in our survey paper. So a moderator is a variable that, if present, changes the relationship between the variables. So for instance, we have an independent variable and a dependent variable in a study, hours of studying and exam score. And we typically would find a positive correlation that the more hours you study, the higher your exam score. The less hours you study, the lower your exam score. But if you consider a third variable here, something called maybe IQ as a moderator, maybe you also give everybody an IQ test and put IQ into this equation, IQ might change this relationship because the more hours someone studied, the higher their exam score. But then when you add IQ in, it just changes the relationship. Maybe in people with higher IQs, you don't really see this relationship between hours of study and exam score. They don't need to study as much to get as high a score. And so it changes the relationship between the variables. So that's what we're thinking about when we're thinking about a third moderating variable, something that changes the positive or negative relationship between the two variables in your study. Now, we can also look at an association between one categorical variable and one quantitative variable. And if you do that, you're doing a t-test, and you'll be doing a t-test in your survey paper as well. And this is a statistic to test the difference between two groups. And we would use a t-test if we do a correlation, which is what we're doing in our survey project, or an experimental study. But in this case, one variable is categorical or nominal, places people in groups, and the other variable is quantitative, which is a number. So for example, if this is an association between where did you meet your spouse and marital satisfaction. So did people meet their spouse online or offline? And then we asked them about their marital satisfaction. Now, this was not an experiment. In an experiment, you would need to randomly assign people to your two groups and then measure marital satisfaction. We are not randomly assigning people to online or offline. This is still a correlational study because we are just observing what's gone on in the environment, what's gone on naturally. We find a bunch of people who met their spouse online and offline, and then we ask them how satisfied they are in their marriage. This is exactly what will be done in your survey project if you're going to compare males and females, or people who are students and people who are not students, any two groups, but you didn't put those people into groups. They are in what we call naturally occurring groups. That's basically looking at what we call a correlational or association data. So for a t-test, your hypothesis is you're going to be comparing the means for your two groups. So if I did a study looking at if people wearing dressy clothes or sloppy clothes are helped sooner in a store, let's say, and we have someone observing in the store and taking down the time it takes for someone to walk in the store, and then someone in the store to say, "Can I help you?" or something like that. And so we're looking at the time it takes to get helped in a store, depending on the type of clothing that someone is wearing. And so let's say for the dressy clothes people who are dressed up, this is how long it took, with a mean of 48.38 seconds. And then we have different people in our sloppy clothes group. This is how long it took for someone to ask them if they could be helped. That's our mean of 63.25. So we've got two groups. We have two means, and we're going to compare the difference between the means using a t-test. But in this case, we weren't assigning people to one of these groups. They just came in the store, either wearing nice clothes or sloppy clothes. And so once you do that, you're comparing the two means. You'll have the standard deviations from SPSS. You'll get a t-value, degrees of freedom, and a p-value, again, less than 0.05 means the groups are different. For a t-test, you'll be doing a bar graph. Here's how long it took the mean for sloppy clothes compared to dressy clothes. And you would say something like, as figure X shows, people wearing dressy clothes were helped significantly sooner than those wearing sloppy clothes. Here's the T. This is degrees of freedom, comes right off your SPSS printout. Here's your T value, again, comes off the SPSS printout, and P, your P value, which if it's less than 0.05, it's statistically significant, and this also comes from your printout. So what makes a study correlational? You need two variables that are both measured, meaning you did not manipulate anything. To do an experiment, you manipulate one variable and measure the other. You manipulate the independent variable and measure the dependent variable. And it's this manipulation that is key to an experiment. We'll be talking about that a little later in the semester, but right now we're on correlational studies, two measured variables. And so when you have a correlational study, you would want to think about the different validities: construct validity, statistical validity, internal validity, and external validity. So let's go through these, right? Construct validity, how well was each variable measured? So did you like how they measured them? Do you like the operational definitions of the measures? If they present these measures, do they have good reliability in terms of their consistency? Did they present test-retest reliability results or inter-rater reliability results? So do you think it's measuring what it's intending to measure, each of the variables? That's what you're thinking about when you're considering construct validity. Statistical validity, you're thinking about the statistics. Is the correlation statistically significant? Did they show you a scatter plot to be sure that the relationship isn't curvilinear or some other kind of relationship, in which case you want to be sure they're using the right statistic, or you're using the right statistic when you're looking at statistical validity? And then we would want to know about what is the effect size, could outliers be affecting the association, and is there a restriction of range? These are important points. And so we'll look at them here. Effect size describes the strength of the association. And basically, the farther apart the dots are, the weaker the correlation. We would say this has a smaller effect size. The closer the dots are together in a line, the stronger the effect size, the larger the effect size. And so we're saying we're better able to predict one variable from the other because these dots are closer together. There's a stronger effect. When everything else is equal, a large effect size is usually considered more important than a small one. But there are definitely exceptions. If you have some kind of disease and there's no real treatment for it, but you find something that seems to be effective, even if it's not got that strong an effect size, well, you might say, I think that's an important effect, even if it's a relatively weak one. Also, you want to look at outliers. Again, this is important. This is why we plot our data first. If you have an outlier is one point that doesn't really fit your pattern of data, it's out here, that can make, statistically make your correlation significant or not significant. For example, if this dot wasn't there, this might be a non-significant, I'm trying to cover that up there. This might be a non-significant relationship, right? It doesn't look like there's much of a relationship there. But with this one outlier, this may end up being a positive correlation, and that might not be really very accurate because this one dot is just sort of throwing all the data off. Or you could imagine this one dot being over here, and now turning this into something that looks like a negative correlation, even though, again, it's just one outlier that might be responsible for that. So you want to look and see why you have an outlier. Is there something different about this data? Was it entered incorrectly as a data entry problem, or is it why is this data point here? And then that's a research methods question of whether you include this data point or not. And then restriction of range is also an issue that you need to see a pattern. You need variability. So if we've got SAT score and college grades here, it doesn't look like there's much of a relationship, but you can see that this is a very restricted range. All we're looking at is individuals who got SAT scores between 1200 and 1600. Those are pretty high SAT scores. There doesn't seem to be very much of a pattern. But if you continued and looked at a larger range, like starting at SAT scores of 400, compare all the way to 1600, then you might start to see a pattern, a positive correlation that people with lower SAT scores tend to have lower GPAs, and people with higher SAT scores tend to have higher GPAs. The pattern starts to emerge when you show the entire range. And then, like I said, about curvilinear relationships, if you actually ran a Pearson correlation on a curvilinear relationship, it would give you a relationship, a Pearson R of 0, because Pearson R is actually looking for a linear relationship between two variables. And so it's going to tell you, no, you don't have a linear relationship here. You actually have a curvilinear relationship. So it's very important to look and see. This is age and years and use of the healthcare system, right? We've got when children are very little, they go to the doctor all the time, they're getting shots, they're getting sick, all of that then kind of levels off, and then at the later ages, people use the healthcare system a lot too. So you do need to plot your data to be sure that you know if you should be using a Pearson correlation coefficient or something else. Internal validity, this has to do with how confident are you about the association between the variables. Can anything else be affecting your results? Are there alternative explanations for your findings? That's the idea of internal validity. If your study is internally valid, then there's no other explanations for your findings. But when you do a correlation and you have not actually manipulated anything, we can't really be sure that our variables, one variable causes the other to change. And you might have heard in your statistics class, correlation is not causation, which makes sense. If you see two variables that are associated, we don't know which came first. This is the temporal precedence problem or the directionality problem. We don't know if variable A caused B to happen or if variable B caused A to happen because we did not manipulate anything, so we don't really know which happened first or which caused the other thing to happen. And then we also don't know if there's a third variable. That's called the third variable problem. Is there a third variable that's causing A and B to both go in their direction? That these two variables aren't related at all, but there's a third variable that's causing both things to happen. So in this instance, if we find a negative correlation between alcohol consumption and GPA, we get a correlation of negative 0.65. So what does that mean? That as alcohol consumption goes up, GPA goes down. Or as GPA goes down, alcohol consumption goes up. Okay, it's a negative correlation, so we have this opposite pattern of results. But you don't know which came first, meaning you can't establish temporal precedence. You can't say that, even though maybe it would make sense to you to say, well, people who drink a lot of alcohol, that causes their GPA to go down. We don't know that to be true because it could also be that having a low GPA causes people to drink. That's possible. They're stressed out, they're afraid they're going to get kicked out of school, and they start drinking alcohol. We don't know which came first in this situation. All we know is that these two variables are related in this negative or inverse way, that as alcohol consumption increases, we tend to see GPA being lower, but we don't know which came first. That's the reverse causality or temporal precedence problem. Sorry, that's I can just move on to also then the third variable. If we know that alcohol use is negatively correlated with GPA, could we think of a third variable that would cause alcohol use to go up and GPA to go down. That's the idea of a third variable. A third variable is something that causes both things to happen. What's something that could cause alcohol use to increase and cause GPA to decrease? And maybe there something like depression. Depression might cause people to increase their alcohol use in terms of self-medicating, and depression may cause decreased GPA because they're depressed, they're not going to class, they're not studying, they have low energy and low motivation, and GPA goes down. And that it's not actually some kind of relationship between alcohol use and GPA, but it's depression that's causing both of these things to occur. And then external validity or generalizability. When you're looking at a study or you're designing a study, you want to think about the research findings and how generalizable they are to the population. How confident that this study will apply to other people and other situations. And how important is external validity? Well, basically, if you don't use a random sample, which we already talked about, random sampling gives you a sample of the population that should be representative. But if you use a convenient sample, right, I only use college students in my research study, you don't have to reject the findings. The findings are still important. And basically, you can accept the results for the group that was studied. Like, yep, this new treatment actually does work in college students. And then the idea of external validity would be used for another study that looks at the same variables, maybe in a different population. So if you find that, yes, your new, I don't know, memory cue, memory treatment works in college students, then maybe the next study would be to try it on, I don't know, high school students. And then you've broadened the external validity because now you've seen it in two groups. And then you say, oh, it worked in high school students. What about in middle school students? Let's try the study there. What about in elementary school students? And so this can be multiple studies to basically get the idea of external or generalizability. Remember, most studies in psychology don't use random samples, but the studies are still important. So it's really a trade-off in that we have highly controlled studies in the laboratory that we'll talk about when we talk about experiments. They have very high internal validity because you really, really control everything in a laboratory study. But they don't have very good external validity. They don't generalize very well. But observational or correlational studies, like our survey project, like our cell phone project, have a lot of real world external validity. They do apply to the real world because you're doing them in the real world, but lower internal validity, meaning we have many more alternative explanations for our data and for our results. Thank you very much.